Hello and welcome to Badger Talks Live, which brings exciting happenings, resources, and talent from your UW flagship campus to the people of Wisconsin and beyond. My name is Mia Williams and I am from Chicago, Illinois. I'm currently entering my third and final year as an undergraduate student at the UW-Madison, majoring in psychology with a focus on the intersection of public health and public policy. I'm also earning a certificate in public policy at the La Follette School of Public Affairs. I've seen the wonderful work the La Follette faculty do, and that's inspired me to apply for grad school at the La Follette School and earn a Master of Public Affairs. I'm pleased to introduce our final speaker for the La Follette School's Badger Talks Live series, Professor Philipp Koellinger. Philipp is an expert in the field of social genomics, or the study of how the interplay of genetics and social environments influences our lives. Today, he will talk about the many ways our genes are linked to our behavior, socioeconomic status, and health. Philipp grew up in Germany and received his BS in economics from the Vrije Universiteit of Berlin. He is both a Master's of Science and a PhD in economics and management science from Humboldt University, Berlin. He is a principal investigator and co-founder of the Social Science Genetic Association Consortium, the BIG BEAR Consortium, and the Externalizing Consortium. His research investigates how genes influence economic behavior and how insights into the genetic architecture of behavioral outcomes can inform social and medical research. He has published widely in the most important journals in his field, including Nature, Science, Nature Genetics, and Nature Human Behavior. He has presented his extensive research in talks around the world. Most recently, Philipp was awarded the La Follette School's very own Daniel Lewis and Genevieve Rusfold Goldie Faculty Fellow Award. His recent column in the Milwaukee Journal Sentinel titled Luck of the Draw, How the Random Results of the Genetic Lottery Can Influence a Host of Your Life's Outcomes, reached millions of readers around Wisconsin. We are very fortunate to have him. Please welcome Professor Philipp Koellinger.
Well, thank you so much, Mia, for the introduction. I really appreciate it. And thank you, Badger Talks team, for hosting this. It's really great to be here. Okay, so let me start by pointing out that we have a wonderful and very productive cluster initiative in social genomics here at UW-Madison that I belong to. The faculty members in that cluster are affiliated with a broad range of academic disciplines and departments across campus, including public affairs, psychology, economics, biostatistics, and population health. And we all share an interest in how the interplay of genes and environments shape life trajectories and health. So if you're interested in this topic, I'd highly encourage you to check out the amazing research that the people in this group are doing and the wide range of classes that they teach. So today, I want to give you a brief introduction on how genes influence our lives. I'll start by giving you a brief reminder about genes. What are they? How do they pass on from one generation to the next? And then we'll talk briefly about heritability. What does this term mean? And importantly, what does it not mean? And probably the most important part is to understand that genes and environments can barely be separated in a really meaningful way. And I'll go over a few reasons why that's the case. And I'll also show you some empirical examples from research on the links between genes, education, income, and health to illustrate to you a little bit what we currently know and also what we don't know. And in the end, I'll contemplate a bit about what our future may look like when genetic data will be much more widely available than it currently is. So let's start with going over the genetic basics. So all our body cells contain a building plan that our cells need to grow and to function. And that set of instructions is called the genome. The genome is the complete set of genetic information in an organism, and that set of information is passed on from parents to their children. Thus, children inherit their genomes from their parents. The genome is stored in a long molecule of DNA called chromosomes, depicted here on the right. And humans have 23 pairs of chromosomes, so 46 chromosomes in total. And if we zoom in, we can see that each chromosome contains a tightly packed string of two connected DNA strands that together resemble a twisted ladder, also known as a double helix. And the rails of that ladder consist of a sugar phosphate backbone and one of four nitrogen-containing bases, which are abbreviated as A, C, T, or G here on this figure on the right. And the bases bind to each other such that A always binds with T and G always binds with C, forming the rungs of the ladder. It's this specific sequence of letters that contains the required information to make different protein molecules that our body needs. And one important empirical fact is that the vast majority of the human genome is actually identical for all humans. And this is what makes us human rather than chicken or onion or some other living organism. But in some parts of the genome, differences in the genetic sequence can be found among people. And those genetic differences play an important role in making all of us unique as human beings. Most cells of our body contain an identical copy of our entire genome in their core, in their nucleus. A big exception, however, are the reproductive cells, the sperms and the eggs, which each contain only one version of each pair of the chromosomes. So these reproductive cells have only 23 instead of 46 chromosomes. As a result, mom and dad both pass down only one version of each chromosome pair to their children, and when the two reproductive cells are combined, the embryo ends up with 46 chromosomes in total. This process is illustrated on the left. And because of this process of combination of the parents' genomes, children genetically resemble each of their parents on average by 50%. And now we may wonder, how can it be that children of the same parents can be so different from each other? How can it be that siblings may look and behave very differently even though they have the same parents and grow up in the same household? An important reason is that siblings are actually not born with exactly the same genome. They are only 50% genetically identical on average rather than 100%. And the reason for that is a wonderful process called meiosis that is also being illustrated here on the left. So meiosis happens when the human reproductive cells, the sperm or the egg, are being produced. The process of meiosis is basically a lottery that draws a subset of our genes and stores that subset in one of our reproductive cells. Specifically, the two copies of each chromosome are being randomly mixed into just one copy during meiosis. And as a result of that, the DNA in each sperm and each egg are a unique random draw of the father's and mother's DNA respectively. And when our reproductive cells combine, they form a unique DNA combination that makes each child genetically unique. The only exception to this rule are identical twins, which occur when a single egg is fertilized and then divides into two separate embryos later on. So the first and maybe also the most important story that our genes tell us is who our parents and who our siblings are. And since our parents got their genes from their parents following the same biological laws, we genetically resemble our grandparents 25% and our great-grandparents 12% and so on. And as a result, our DNA contains traces that lead back into our long-term history and that link us with ancestors we had hundreds or even thousands of years ago. And they also tell us to which extent we are related to other people on the planet, and they allow us to estimate when we shared our last common ancestor with another person. In this way, genes are a window into our family history and the history of our species. But that's, of course, not the whole story that our genes can tell us. They can also tell us a lot about different traits and predispositions that we carry in us, which brings us to the topic of heritability. So heritability is probably one of the most misunderstood scientific concepts out there, and those misunderstandings have shaped the discussions about heritability in really unproductive and unfortunate ways. So let's make sure we understand what we are talking about when we use the term. This is also important for understanding how genes can influence us, what this means, and importantly, what this does not mean. So in a nutshell, heritability is a measure for how important inheritance is for a particular characteristic of people that we're interested in. So those characteristics could be anything that makes people different from each other, such as their body height, their hair color, their personality, their education, or a particular health outcome. So scientists call these characteristics traits. And scientists have studied the heritability of thousands of different traits in the past few decades. More precisely, heritability is a statistical concept that measures how well the differences in people's genes account for differences in their traits. As a result, heritability puts an upper bound on how much genes contribute to differences between people. But, and this is important, just because a trait is heritable does not mean that the environment is irrelevant for that trait. And maybe counterintuitively, the environment can still be hugely important, even for traits that have an estimated heritability of 100%. So to illustrate that, let's assume short-sightedness would have a heritability of 100%. Even if that would be the case, it would still matter a lot if you live in an environment with easy, affordable access to eyeglasses or not. And if you happen to live in such an environment, it would still be your personal choice if you would wear these glasses or not. In other words, just because sight or nearsightedness is highly heritable does not mean that we as individuals or we as a collective society cannot change or influence that trait. Another important realization is that heritability estimates are always specific to the population sample that is being studied, and researchers typically select samples from a relatively narrowly defined geographic area and period of time to do that. And this implies that environmental differences that vary between groups or over time and that influence the outcome of interest are typically not taken into account by these estimates. So for example, body height has substantially increased throughout the Western world in the past 100 years, although height is one of the most heritable traits that we know. The reason for that increase in body height is not that our genes have changed dramatically in the past 100 years. They actually haven't. Instead, it's because we have better nutrition, better hygiene, and better medical care now than we had 100 years ago. In other words, the environment for height has actually improved substantially over time. And as a result, most kids who grow up in the Western world now are taller than they would have been if they were born with the same genes 100 years ago. It's also important to realize that heritability is not a natural constant. It's a population-specific parameter that may vary across time and environments, and that may change quite substantially as a result of changes in the environment. So for example, the heritability of a trait such as educational attainment could be much higher in a meritocratic society where the best students end up going to the best schools, compared to a society where kids are just randomly sorted to schools. In fact, the heritability of a trait can be changed or even entirely caused by a specific environment. It's equally important to understand that many, if not most, genetic effects can act via environmental and behavioral channels. The genes we're born with, they influence our body type and they pre-wire our brains in certain ways, but our bodies and also our brains, they react to our environment, including the feedback that we're getting from others and the social circumstances that we grow up in. To some extent, we even create or we choose our own environments, for example, by making choices about who we are friends with and how we spend our time. And all these environmental conditions, they also shape us and they rewire and retrain our brains over time. And this can lead to important environmental feedback loops that either emphasize or de-emphasize small genetic differences between people over time. So for example, imagine two siblings who are quite similar at the outset, but one of them is genetically slightly more interested in science, while the other is genetically slightly more interested in sports. If parents, friends, and teachers of the two siblings emphasize these differences, for example, by encouraging each sibling to focus on their particular interest, the differences between them in terms of aptitude, character, and experience can end up being really substantial over time. Thus, the small genetic differences between these kids can influence their behavior, their experiences, and possibly also their education, their jobs, and their entire adult life via environmental channels that we can influence. And as a result of all of that, heritability does not imply any type of biodeterminism, and it doesn't limit our freedom of choice as individuals or as a society in a strict way. Heritability estimates are consequently also not informative about the nature of differences between groups or over time, and it makes no sense to cite heritability estimates as some sort of natural justification for inequalities. They're not. Okay, so with these clarifications and caveats in mind, let's look at some heritability estimates from the literature. So the estimates I'm reporting in this table here are mostly from a meta-analysis of all twin studies that were carried out before 2015. So there are some diseases and disorders that are caused by a specific genetic mutation, for example, Huntington's disease. And these disorders have a heritability of basically 100%, and they're caused by only one specific gene. But that doesn't mean that these diseases cannot be treated. Also, monogenetic disorders tend to be very rare in the population and typically affect less than 1 out of 1,000 people. The vast majority of traits that are common and that we care about have a heritability that is much smaller than 100%. And these traits are what we call genetically complex. That means that the heritability of these traits is not due to one or a few genes that have huge effects, but rather the heritability is due to the influence of hundreds or thousands of different genes, each of which has only a tiny influence on the trait. Examples are body height and bipolar disorder, but also brain structure and blood pressure. These are all genetically highly complex traits that also have pretty high heritability estimates of 50 to almost 80%. Interestingly, many traits that people may normally consider as social are also heritable. For example, educational attainment, alcohol abuse, depressive episodes, how high people score on personality dimensions such as openness and neuroticism. Again, this doesn't mean that personality, education, or alcohol abuse are determined by our genes or that we can't do anything about it. Instead, this only means that within the environments that scientists have studied until now, genes contribute something to the observed differences among these traits for reasons that often work via environmental and behavioral channels that we may be able to influence. So, if we would know the genes that underlie these heritability estimates, we may be able to learn something interesting about these traits and how they're linked with one another. And if we would have access to our own genetic data, we may also use these insights to learn something interesting about ourselves. So this brings us to the question, how do scientists find genes that are associated with a particular outcome? So the figure here gives an overview of how such studies usually work nowadays. At the beginning, scientists collect data, often in population samples that allow studying more than one potential outcome of interest. And after study participants have been informed about the goals and methods of a study, and after they have consented to participate, one can then take saliva or blood samples to get a tiny bit of DNA for analysis from these people. The DNA is then processed in a laboratory and the extracted genetic information is stored in a data set. The next step is what we call quality control, where the scientists try to identify and fix anything that might have gone wrong during the data collection process. At this stage, the genetic data are also used to calculate the genetic ancestry of the study participants. And in the vast majority of studies, scientists focus on only one ancestry group when they try to find genes that are associated with a particular outcome. So this focus on just one ancestry group limits the risk that the genetic associations that are found merely reflect artifacts due to differences in ancestry, culture, or other relevant living conditions between different ancestry groups. In reality, this step often implies that the genetic analyses are only carried out in individuals of European descent, because in most cases, that's the ancestry group for which most genetic data has been collected. And as a result of this, the vast majority of genetic association studies are conducted on samples of European ancestry only, and they have limited application to other ancestry groups. After this data cleaning process, scientists scan the entire genome for locations where genetic differences between people co-occur with differences in the outcome of interest. The results of these so-called genome-wide association studies, or GWAS, are often shown in a plot that looks like panel E here. So there is a dot for each genetic marker in that plot, and the genetic markers are ordered on the horizontal axis according to their physical location on the genome, and on the vertical axis according to how confident we are that that this particular genetic marker is associated with the outcome of interest. So we call these figures Manhattan plots because they resemble the skyline of New York, and all the towers in such a plot that are higher than that red line point to a region of the genome that is associated with the outcome. So often the effects of each individual gene on an outcome are so small that scientists need really large samples to identify these genetic signals reliably. Usually this means that we conduct GWAS in more than one sample, and we summarize the results from all samples in a so-called meta-analysis. And to make sure that our discoveries are not just statistical flukes, we conduct so-called replication studies where we check if the findings from our discovery sample can also be found on a different independent sample that wasn't part of the original discovery. And once that has happened and we convince ourselves that our findings replicate, different kinds of follow-up analysis can be done that can help us to better understand what these statistical associations actually mean. So following this process, many robust, replicable genetic associations with hundreds of different traits have been reported in the past few years, including several large-scale studies on measures of socioeconomic outcomes and their links with genes. So for example, my team has been involved in and led a number of really large-scale genome-wide association studies on educational attainment and samples of people with European ancestries. And the number of regions in the genome that we identified as being related to educational attainment increased dramatically in the past few years, from just 3 regions in 2013 in our science paper, which is on the top left, to 74 regions 3 years later in our Nature paper on the top right. And then finally to more than 1,000 regions in 2018 that we published in a journal called Nature Genetics. And that's seen on the, switching back. Yes, and that largest study is seen in the bottom. So the number of genetic regions that we found to be associated with education attainment did not grow because of some fundamental changes in the importance of genes for education. Rather, the sample sizes that we could investigate grew dramatically over time. So while our first study in 2013 was based on about 100,000 individuals, the latest version in 2018 was based on more than 10 times as many study participants, actually more than 1.1 million in total. And with growing sample sizes, scientists can gain more reliable and more precise knowledge about the relationships they're interested in. In this example, our 2013 study only identified the tip of the iceberg, while our later studies got better and better in identifying all the various regions of the genome that are linked with education attainment. So robust genetic associations for other measures of socioeconomic outcomes have also been reported, for example, for income and for neighborhood quality. And it became increasingly clear over the past few years that the genetic factors that are linked with socioeconomic outcomes are also associated with a variety of environmental factors, such as which neighborhood people grew up in. Thus, these genetic associations also tell us a story about relevant environmental factors. So let's zoom in on the latest genome-wide association study on education attainment, which is basically, so education attainment is the highest degree that people have obtained in school. And in this latest study, we found almost 1,300 locations in the genome that are robustly linked with education attainment. But even though these associations are robust in statistical terms, the effects of each genetic region considered by itself are really, really tiny. So each genetic region that we see here in this plot explains less than 0.1% in educational attainment. In other words, there is not one gene for educational attainment. Rather, we're looking at a genetically highly complex trait where literally thousands of genes are involved in education attainment for some reason. And Each of them contributes only a tiny bit to the variations in schooling outcome that we observe. Okay, another way to look at the results of such genome-wide association studies is by aggregating the estimated effects of all the genetic markers in a so-called polygenic index. In the figure here, you see one dot for each individual in a sample that wasn't part of the original GWAS on education attainment. On the horizontal axis, you see the values of the polygenic index, and on the vertical axis, you see education attainment measured in years of schooling after we've controlled for differences in schooling between people that can be attributed to sex, birth year, or ancestry. The blue line is called the regression line, and it summarizes the relationship between the polygenic index and education attainment in the sample. The positive slope of that blue line indicates that people with a higher polygenic index are, on average, more likely to get higher education. More specifically, the polygenic index explains roughly 11% of the observed differences in years of schooling in this particular data set. And thus, this particular index tells us a lot more about education attainment than each genetic variant by itself, because it summarizes all the information from across the genome. In fact, this particular polygenic index for education attainment is one of the most accurate genetic indices that are currently available. So it's a pretty good benchmark for seeing what can and cannot be done with this type of information at the moment. So one thing you could do out of curiosity is to check where your own polygenic score would fall in this distribution. And this is actually not that difficult to do nowadays. So anyone can have their genome measured, for example, by a consumer genetics company. And many of these companies allow you to download your own genetic data. Then there are also web services available that interpret genetic data and that allow you to construct polygenic indices for different traits with your own data. So this may sound futuristic, but this is already possible for several years, and I personally find it pretty fun to do. But how much would you learn about yourself if you constructed your own polygenic index for education attainment? So let's look at this plot a little bit more carefully. So let's say your own polygenic index is a little bit above average, let's say 1 standard deviation above the average. So the blue regression line tells us that people with that value of the polygenic index complete on average 14 years of schooling, which is equivalent to going to college for 2 years. But you also see that education outcomes have been all over the place for people with that particular polygenic index value. In fact, the range includes everything from high school dropouts to people with PhDs. So although this analysis tells us something meaningful about what we would expect to see on average, it is not particularly useful for telling us something accurate about yourself or any other particular individual. So in other words, even though the polygenic index for education attainment is one of the most powerful and accurate ones that is currently available, it is still not nearly precise enough to be useful for individual level prediction. But it is very useful as a tool for scientists to understand mechanisms that are linked to inequality and socioeconomic status and also in health at the aggregate level. And it has been used for that purpose in many different studies after we published our original results. So another very useful thing scientists can do with genome-wide association study results is to compare them for different traits, to see whether the genetic underpinnings of different traits actually resemble one another. And the results of these comparisons can be summarized in a single number that is called the genetic correlation coefficient. And maybe surprisingly, with GWAS summary statistics, we can calculate genetic correlations between pairs of traits, even if these traits have never been measured in the same sample in the same people. So this allows us to see connections between traits at the genetic level that we didn't know before. So as an illustration, this figure here summarizes genetic correlations between educational attainment and a variety of other traits that were studied in other GWAS studies. So we see that education-associated genes are also associated with many other traits across the lifespan. So for example, they're also linked with higher birth weight, larger infant head size and higher childhood IQ, but also with lower body mass index and lower obesity, a lower chance to experience depressive symptoms or neuroticism, and a lower chance to be a smoker. And towards the end of the life cycle, we see that education-associated genes are also linked with a lower chance for lung cancer, Alzheimer's disease, and a higher chance to live longer. So these positive health implications of education-associated genes are marked in green in this figure. However, the effects of education increasing genes are not universally positive. They are also linked to reduced reproductive success, so measured by fewer children that were born or higher age at first birth, as well as an increased risk for several psychiatric disorders, including autism and bipolar disorder. So these findings underscore how tightly social scientific outcomes such as education are linked with health. The most important lesson here is that it's often the same genes that influence health and socioeconomic status. There is no clear line between the genetic architecture of behavior or socioeconomic outcomes on the one hand and health on the other. Education and health are related to each other, both genetically and in terms of lifetime outcomes that we can actually observe. Now let's remember that children genetically resemble their parents by 50%. The genome association studies that I was talking about earlier usually do not control for the genes of the parents. So therefore, it could very well be that some of the genetic associations we've seen are actually working via environmental conditions that the parents create for their children and that are linked to the parents' genotypes. And these indirect genetic effects are sometimes referred to as genetic nurture, and the genotypes of the parents shape how the child grows up and may therefore have an indirect impact on the child that operates via the parents' behavior. And recent studies using genetic data from parents and their children actually suggest that these indirect genetic effects account for about half of the association between the polygenic index for education attainment and the schooling outcomes of the offspring. Thus, perhaps counterintuitively, these sorts of genetic study designs allow us to gain a fresh look at the importance of parenting, and they remind us that genotypes and environments are forever tightly intertwined because parents pass down their genes as well as their environments to their children. So one way how we can separate the direct and indirect effects of genes better is by looking at the genetic differences between siblings. And as I said at the beginning, siblings are on average 50% genetically identical. And the genetic differences between them are the result of a genetic lottery that happens during the process of meiosis. As a result of the genetic lottery, one sibling will, by pure chance, have a slightly higher polygenic index than the other, and we can look at the consequences of this genetic lottery by examining observed differences between these siblings later in life. So we did that using GWAS results for income that we applied to construct a polygenic index in a holdout sample of about 35,000 biological siblings in the UK Biobank, taking advantage of digital health records, self-reports, and other assessments that we had available in that data set. And our results show that random differences in genetic endowment for income between siblings translate into inequalities in health and socioeconomic status later in life. In particular, the sibling that won the genetic lottery for income was more likely to get a college degree, a higher wage, and to live in a wealthier neighborhood than the sibling that lost this genetic lottery. Furthermore, the sibling who won the genetic lottery for income was also less likely to get sick or obese later in life, and was more likely to have healthy lung function and lower blood pressure. So all of this illustrates real-world consequences that tiny random differences between people can have, even though nobody chooses to be born, even though nobody chooses their parents or the genes that they endow us with. The results of that genetic lottery, the genes that we're born with, they play an important role in how well we are doing later in life, both in terms of our health and in terms of our social and economic achievements. So The results of such genetic studies, they challenge the view that people deserve everything that happens to them. I think it's just difficult to argue that we should take full credit or full blame for the outcomes of the genetic lottery. That's a lottery that neither one of us could influence or decided to participate in the first place. Instead, the results of modern genetic studies can be interpreted as a strong argument in favor of solidarity, humility, and sympathy for others. They can even be used to support policies that are trying to level the playing field, as my colleague Paige Harden from the University of Texas Austin forcefully argues in her new book. Now let's speculate a little bit about what the future may look like. One thing we know for sure is that genetic data is becoming really cheap. It's already possible nowadays to genotype a person for as little as $20. That's cheaper than conducting a telephone interview and much cheaper than most medical examinations. As a result, tens of millions of people have already been genotyped, often to participate in research studies, but a lot of people also paid companies to get genetic information about themselves. So with the increase in the availability of genetic data, researchers keep learning more and more about which genes are linked to which outcomes. It will also be possible to develop better, more precise prediction algorithms that will include genetic data and that could be used for a wide range of applications. And importantly, once you have information about somebody's DNA, it is possible to obtain information about any heritable trait that person might have, to the extent that we know which genes are underlying this heritability. Thus, for just $20 or even less, we may be able to extract a whole range of interesting information about a person and their potential future. And this is such a strong value proposition that it's easy to imagine that the collection of such data could soon be considered as standard. In some countries or some regions and some families, all newborns may be genotyped at birth or even earlier, allowing potential insights into the traits these kids are likely to develop over their lifetime. At this moment, just to be clear, at this moment, this is still science fiction. But these developments are already happening to some extent, and they proceed at a rapid pace. On the one hand, there are many possibilities that these developments will bring with it. For example, when genetic data is available for everybody, and when we have good predictive algorithms that give us insights into the disease risk that people carry, it would be possible to have much more personalized, targeted, and effective medical care. This would start with focusing preventive care and screening on those people that have the highest risk of developing a specific disease. And it may also involve personalized treatment plans for those who got ill. Consumer genetic companies may be able to produce better, more precise reports about a growing number of traits, allowing their customers to learn a lot of interesting things about themselves and their families. Scientists will be able to use genetic data in a lot of different ways that will allow them to improve the quality and the efficiency of their studies. For example, by using polygenic indices to approximate traits that they do not measure directly. But then there's also other applications that may not be so benign. So for example, insurances may be able to offer discounts to people who have a lower risk profile. And employers may want to use genetic data to improve the matching between employees and jobs. And finally, genetic information could be used to improve the health of the next generation, for example, by eradicating monogenic diseases using assisted reproductive technologies such as in vitro fertilization after genetic screening. And all of that will be, and to some extent already is, technologically possible to some extent. But also, all of this comes with big risks, including the risk of genetic discrimination. For example, would you really want your health insurance to know that you have an increased risk for cancer? Probably not. Similarly, would you want your employer to compare your genes with those of other people to determine who will get that job that you really wanted? Probably also not. Furthermore, we've seen how unreliable genetic predictors currently still are. Would we really want to use such an inaccurate measure as the basis for any important decision? I also mentioned at the beginning that the vast majority of genetic association studies is currently being carried out with data from people of European ancestries, and that the results of these studies do not fully carry over to other population groups. So to the extent that the results of these studies may have clinical or other value, the benefits would be limited to a minority of people on this planet, potentially increasing inequalities further. And all of this also raises many questions about data security and privacy. So keep in mind that the genetic information we carry in us also tells us a lot about our closest biological relatives, for example, our parents, our siblings, and our children. So whenever we reveal our genetic information to somebody else, we are inevitably also revealing some genetic information about our biological relatives, even though they may not have been asked and they may not have provided consent for sharing that information. And once somebody's DNA is stored in a database, how do we ensure that this information doesn't leak out, get into the wrong hands, and is being used against us? Is privacy perhaps an illusion in such a future world? All of this raises many ethical dilemmas and questions that we as a society need to think about. So we as a society will have to discuss and to agree upon what type of society we want to live in the future. Who should have access to our genetic data, under which circumstances, and for what purposes? Different people in different societies may come to very different answers to these questions, and we may see very different policies about this being implemented across the world. But whatever these policies will be, they will shape our future and the lives of our children and grandchildren. So in summary, our genes influence nearly every trait that we care about, but most traits are influenced by many genes with small effects, and many of these genes influence many different traits. Which genes matter for a particular trait may in principle vary across time, place, and population, for example, as a result of different environmental conditions. As a result, it really doesn't make sense to say that there is a gene for education, income, or personality. Things are actually much more complex in reality. I would even argue that the old nature-nurture debate is entirely misleading because genes and environments depend on each other and they co-evolve with each other over time. So they are forever tightly intertwined and they both matter. But one thing we know for sure now is that the genes that we're born with have some degree of influence on who we are and what happens to us in our lives. Thus, genetic luck matters and influences nearly every dimension of our lives. And I'll leave it here and open it up for discussion.
Philip, thank you so much for sharing that important research and information. And it's apparent that you've given us information to help us better navigate what our gene structure is telling us for the rest of our lives, and how maybe if we have that information, we can kind of prepare for that, and maybe environmental factors can influence that. But even more importantly, Philip, we now have the building blocks to better understand how to successfully navigate Thanksgiving dinner with extended family.
Okay. Well, I'm glad I helped you on that front.
Well, hello, everybody. Fran Paleo-Moyer, Badger Talks producer. We do have quite a few questions in the chat, as you can imagine, on such an interesting topic. So a question here from Paige Bazan. The social genomics researchers you listed earlier come from a variety of professional backgrounds. What kind of academic or career paths would you recommend for those interested in studying this?
Oh, so I actually have a very simple answer to that. I think really the gateway into that type of research is just a sound understanding of statistics, and you can get that in a lot of different disciplines, in economics, in statistics, in biostatistics, in medicine, and psychology. There's many different ways how you can get into that. And then ultimately, it's just a matter of applying these statistical techniques to a particular type of data, in this case, genetic data.
Kind of on top of that subject, how did you get interested in this?
Oh, yeah, that's actually a pretty long story. But the short version is that I've always been interested in genetics. And then when I was basically in my tenure track job at the University of Rotterdam back in the days, I was in the economics department. I realized that that university had a medical school that had a gigantic genetic data set. So my colleague and I, we went over to the medical school and we asked them if we can work with them. And they did something really stupid. They didn't say no to us. So we ended up studying genetics.
Very interesting. So Brittany Mitchell posts, what research topics will you be studying in the next few years?
So that's a great question. So we have a number of studies that are currently in the making that are related to other measures of socioeconomic outcomes, for example, income and also family wealth and the type of neighborhoods people live in. We will definitely keep working on a variety of mental health related traits, also traits that are related to impulsivity. And I also have a number of studies that start looking at one of the missing pieces in the puzzle, which is the brain, right? So a lot of the things that we've seen are genetic effects on behavior that must involve the brain in some way. And we've started incorporating brain images in these genetic studies, and we're learning a lot about the brain that way.
Very cutting edge, thank you. Ann Schaefer posts, can you talk about the relationship between your research and public policy, and what might policymakers do differently in light of what you and other social genomic researchers are learning about the interplay between genetics and environment?
Right. So I think probably for policymakers, the biggest takeaway message is that this genetic data is out there, it's growing, it will become more and more available. We will learn more and more things about how these genes are linked to all different types of outcomes. And we need to have an informed, open debate in society about how we're going to deal with that situation, with that new data, with that new information. How are we going to integrate it into our societies? And I think that's really the big debate that needs to happen relatively soon. And I don't have a clear answer about, what is going to be the best way of dealing with that, and what are the decisions that we should make. But I think we cannot run into this technological progress that we're seeing with folded eyes, right? So we cannot pretend that this is not happening and that it's not relevant. It is relevant and it will influence our lives.
Good point. Let's see. We have a question from Tiago Ribeiro. Was there an overlap between genetic markers for educational attainment measured by GWAS and in the study with siblings?
Sorry, I'm not really sure I understood that question. So can you maybe repeat?
Yeah, sure. Was there an overlap between genetic markers for educational attainment measured by GWAS and in the study with siblings?
Okay. Yeah. So I guess this is the question whether these results that we found in the GWAS study, whether they also were found in samples of siblings. So the answer to that question is broadly yes. So we do see that the same pattern of results that we see in a population sample emerges when we compare siblings with each other and we look at their education outcomes and also their income.
Great, thank you. Kelly Bauman posts, how are these data confounded by people who may not have received an official diagnosis, say for celiac disease?
Yeah, so when we look at outcomes like education attainment, so this is not really a case control thing. So there isn't really that somebody is diagnosed with something. So this is based on self-reports. But the problem is still the same that you're referring to. It's measurement error, right? So there's going to be some degree of measurement error, and what this does is it basically makes it more difficult for us to find genetic associations with the traits that we're interested in. If anything, this type of classic measurement error or misclassification, it actually makes it harder for us to find genetic effects.
Thank you. Bonnie McRitchie posts, you mentioned that the genetic data you work with is primarily from people of European descent. Do you think that will change sometime soon? Will you be able to study genetic data about other groups?
Oh yeah. So I'm absolutely certain that this will change at some point, but this is going to take some time, and the extent to which this is happening largely depends on how much money is being spent on genotyping efforts and to which extent people are actually willing to participate in such studies. So what we're seeing at the moment is that in a lot of Asian countries, there is a lot of interest in genetics, and there's huge investments into collecting genetic data, for example, in China and in Japan and in Korea. And we're also seeing some efforts to start genotyping people from Africa, and this is really where most of the genetic diversity that we have in the human population is present, and this is also the area that is most difficult to actually cover comprehensively in terms of genetics. So I think it's going to take us quite a while until we have a really good idea about not only what different types of genetic variants are there, but also what effects they have and how they may differ from the effects that we see in other populations. But I'm really sure that this is only a matter of time until we will be able to extend these types of analysis to other population groups.
Philipp, thanks so much for sharing your important work with us. We've been just really in awe of hearing from all of the speakers we've showcased here on the La Follette School of Public Policy series, and your work is also a genuine testament to that. So thank you for your good work.
Very welcome.
Thanks for tuning in. Please join us in the month of September. We're going to be featuring the Mead-Witter School of Music, taking a little bit of a turn toward the arts. The next talk will be Tuesday, September 14th at noon. And we're going to be kicking it off with Assistant Professor of Trumpet, Jean Lorenz, and she's going to be talking about her unique approach to keeping audiences engaged through interdisciplinary collaboration. We'll continue this series September 21st with Johannes Wallman, Associate Professor of Jazz Studies, Friday, September 4th with the Professor of Horn, Dan Grabois, and Tuesday, September 28th, the Director of the School of Music, Susan Cook, will be giving us a tour of the phenomenal New Hamel Music Center, so don't miss it. Please visit us at badgertalks.wisc.edu, where you can see the upcoming schedule of live talks, and we have posted through the end of the school year, so please do take a peek out there. Sign up for our email list where you're the first to know about upcoming events. Consider a donation to Badger Talks. We are supported by a grant. And search the roster of over 400 UW faculty and staff we have signed up to give talks in person and virtually around the state. We look forward to hearing from you again. Thanks for tuning in, and we'll see you in a couple of weeks. Thanks much.